Welcome to the Biology Brief channel. Today we are dealing with the last part or third part of MCQ discussion of the chapter Molecular Base of Inheritance. Today we are going to discuss about 15 MCQs regarding the topics regulation of gene action, human genome project and DNA fingerprinting. So this is a short session. We are moving to first question. First question. In eukaryotes, the regulation of gene action could be except at the following except. In eukaryotes, the gene action is regulated at a transcriptional level, processing level, translation level and transporting level. Transporting level. There are 4 levels of regulation of gene action in eukaryotes. So our answer to this question is fourth option, replication level is a wrong option. So that is our answer. Here in transcriptional level, the stage of primary transcription formation, gene action is regulated. During mRNA splicing or mRNA processing, the gene action is controlled. During the formation of polypeptide chain synthesis, there is regulation, and when mRNA is transported or the primary transcript is transported from the nucleus to cytoplasm, there is also regulation of gene action. We are moving to next question. Again, regulation of gene expression. In regulation of gene expression, the regulatory protein that acts positively is called. The regulatory protein which acts positively is called an activator. Sometimes the regulatory protein will act negatively. Then it is called repressor. What about next question? In lactoperon, lactose molecule acts as, you know that, in lactoperon, lactose molecule acts as inducer. So you can strike out second and third options. First option, inducer which binds with operator gene. Wrong answer. Inducer will not bind with the operator gene. The repressor protein binds with operator gene. Fourth option, inducer which binds with the repressor protein. This is the answer to this question. Inducer lactose. Lactose will bind with the repressor protein and the repressor protein becomes inactive and it cannot bind to operator gene and the lactoperon is switched on. You have studied. What about next question? An experiment was conducted to understand the regulation of gene expression in Escherichia coli. The bacteria were grown in medium containing lactose. There is lactose in the medium. Listen that. No glucose. Which of the following outcome is expected? First option. The repressor protein remains bound to the operator, blocking RNA polymerase and preventing transcription of operon. Wrong option. The repressor protein cannot bind to the operator when lactose is there. Lactose being inducer, it binds with the repressor and makes it inactive. So wrong option. Second one, the lac repressor protein gets inactivated by lactose. Correct answer. The lac repressor protein combines with the lactose and becomes inactivated and it cannot bind to the operator site, allowing RNA polymerase to bind the promoter and initiate transcription of operon. It is the answer to this question. Second option is answer to this question. The third option, the presence of lactose has no effect on the lac. Wrong option. Both glucose and lactose are required simultaneously. It is also a wrong answer. So our answer is second option. Next question. Gene regulation governing lac operon of Escherichia coli that involves a lac-I gene product is. We know that lac-I gene product is repressor protein. Repressor protein act negatively on gene action. Negatively on, but its action is negative. So you can remove second option from your list. Then what about first option? Negative and repressible because repressor protein prevents transcription. It is the correct answer to this question. First option is correct answer to this question. It is called negative because it prevents transcription. It repress the process. So first option is the answer. Negative and inducible, not inducible. Option 3 and 4 are also wrong options. So we are going to next question. The Escherichia coli grown in cultural medium containing no lactose in the medium. The lac operon system will remain switched off completely, remain active at very low level, normal level, very high level. Wrong options. When there is, even though there is no lactose in the medium, the lac operon system remains active at a very low level, and a low level of permease enzyme will be formed. This permease allows the lactose when it is present in the future. Initially the lactose will be entering into the cell by the formation of a little amount of permease. So the lac operon remains active at a very low level even though there is no lactose. Second option is our answer. Next one, seventh question, matching question. Structural gene. We know that structural gene codes for polypeptide chain. Operator gene is the binding site for repressor protein. Promoter gene. In this promoter gene, RNA polymerase will bind to the promoter gene. Regulator gene codes for repressor protein. For option A3, B1, C4, D2. Fourth option is easy question. Fourth option is our answer to this question. Next question, eighth one, statement type. Less than 20% of the human genome code for protein. Carefully read the question, such questions, and reach at the correct option. Less than 20% is a wrong data. Less than 2% of the human genome codes for proteins. So statement 1 is a wrong one as such. What about statement 2? Largest known human gene is the one codes for dystrophin protein and has 2.4 million base pairs. It is a correct statement as in the textbook. So statement 1 is wrong, but statement 2 is correct. Fourth option is answer to this question. Fourth option. Moving to next question. Which of the statement is not correct? You have to select the statement which is not correct. Not correct. First statement. Less than 2% of the genome codes for proteins. Correct one. Correct one. Then chromosome number 1 has fewer genes and Y chromosome has most genes. This is a wrong statement. Y chromosome has fewer genes. Y chromosome, minimum genes are present in Y chromosome. The data is 231 genes are present in Y chromosome. Chromosome number 1 has most genes. The number is 2968. 968 genes are present in chromosome number 1 and Y chromosome has fewer genes. The number is 231. The second statement is wrong one. What about that? The repeated sequences make up very large portions of the human genome. Correct statement. The repeated sequences or the repetitive DNA or satellite DNA make up very large portion of human genome. Correct statement. The human genome contains 3164.7 million base pairs. It is also correct statement. So 1, 3, 4 are correct. Here asked not correct, which is not correct. Our answer is second option. Next question. Select the incorrect statement. Here again incorrect statement is asked about the variable number of tandem repeats or VNTRs. VNTRs, variable number of tandem repeats. It belongs to a class of satellite DNA. The statement is correct one. A small DNA sequence is arranged randomly in many copy numbers. A small DNA sequence arranged randomly in many copy numbers is also correct one. The size of VNTR varies from 0.9 to 50 kb. It's a wrong statement. 0.1 to 20 kb. The size of VNTR is actually 0.1 to 20 kb. Third option, third choice is wrong one. It differs individual to individual in a population except in the case of identical twins. In the case of identical twins, the VNTR is same. VNTR pattern is same. But in the case of individuals in a population, it varies individual to individual. It is a right option. So our answer to this question is option number 3. Then moving to 11th question, statement type. Read the following statements regarding DNA fingerprinting. Here the statement is given regarding DNA fingerprinting. First one, the technique initially was developed by Alex Jeffreys. Correct statement. Alex Jeffreys developed DNA fingerprinting initially. The technique involves Western blotting hybridization using radio-labeled VNTR as probes. For the identification of DNA segments, Western blotting is not used. Here, Southern blotting is used. Southern blotting. So the statement is a wrong one. You have to add Southern blotting to make the statement correct. As such, it is a wrong one. VNTR belongs to microsatellites. It is also wrong one. VNTR belongs to mini satellites. Mini satellites. The satellite DNA are divided into mini satellites, microsatellites, and etc. Satellites. Here VNTR belongs to mini satellites and the statement 3 is also a wrong one. Fourth one, detection of hybridized DNA fragment is done by autoradiography. The statement is also correct one. Only two statements are correct. 1 and 4 are correct. Here asked incorrect statements which are incorrect. Second and third. Our option to answer to this question is third choice. Second and third options are wrong here. 12th question. The probability of assertion, the probability of DNA variations would be high in non-coding DNA sequences. The probability of DNA variation is also called DNA polymorphism. DNA polymorphism. The DNA polymorphism is high in non-coding DNA sequence. The assertion is correct. Mutations are the reason for this DNA polymorphism. Mutation in non-coding DNA sequence may not have any immediate impact in an individual's reproductibility and keep on accumulating generation after generation. It is also correct and it correctly explains assertion. So our answer to this question is option 1. Both A and R are true and R is the correct explanation of A. You have to read carefully and arrive at a correct answer. Next question, 13th one, this is also assertion reason type. Assertion, the repetitive DNA are separated from bulk DNA as different peaks during density gradient centrifugation. Here the DNA, repetitive DNA are separated from bulk DNA by the technique density gradient centrifugation. Assertion is a correct one. Reason, the bulk DNA forms a minor peak and other major peaks are referred to as satellite DNA. It is a wrong one. The bulk DNA forms major peak and other minor peaks and the satellite DNA forms other minor peaks. Bulk DNA forms major peak and satellite DNA forms minor peaks. The reason is wrongly given here. So our answer is A is true, R is false. Third option will be answer to this question. Moving to next question. Select the wrong statement from the following. Read carefully. First one, VNTR belongs to a class of mini satellite. We have mentioned earlier this correct statement. VNTR belongs to mini satellite. Human Genome Project was coordinated by US Department of Energy and US National Institute of Health. The statement is also correct one. Satellite DNA do not code for proteins. Satellite DNA, repetitive DNA, do not code for proteins, correct? But they form a large portion of human genome. It is also very correct. Fourth statement, automated DNA sequencers that worked on the principle of Frederick Sanger were not used for sequencing the DNA fragments HGP. HGP means Human Genome Project. The statement is wrong. We have to remove not from this statement to make it correct one. Automated DNA sequencers are used during Human Genome Project. Our answer to this question, here asked wrong statement. Here the answer is fourth option. Then we are moving to our last question. Which of the following statements are correct? Here the statements are given regarding polymorphism. First you have to understand what is polymorphism. We know that DNA polymorphism, it is due to the varied copy number of nitrogen-based sequence or sequences in DNA segment from chromosome to chromosome in an individual. This DNA polymorphism may range from single nucleotide change to a very large scale changes in the repetitive DNA sequence. Then the DNA polymorphism is at genetic level variation. There are genetic level variations and they are present in the non-coding DNA sequences. You have to know these facts before answering to the next question. Then let us move to the statements. First statement, polymorphism in DNA sequence is the basis of genetic mapping in human genome. Polymorphism DNA sequence are markers, identifying markers, so they are used for mapping human genome. Correct statement. Polymorphism is not the basis of DNA fingerprinting. It is a wrong statement. Polymorphism is the basis of DNA fingerprinting. VNTRs are a type of polymorphism. Third statement, polymorphism, that is variation at genetic level, arises due to mutation. Very, very correct statement. Polymorphism are variation at genetic level and they arise due to mutations. Satellite DNA has high degree of polymorphism. Satellite DNA or repetitive DNA or DNA polymorphism has high degree of polymorphism. Correct statement. Polymorphisms are not inheritable from parents to children. That is a wrong one. Wrong one. We have to remove not from the statement to make it a correct statement. Polymorphisms are definitely inheritable from parents to children. It is a wrong one. So here asked for correct statements. Correct statements except B and E, that is A, C and D. Fourth option is our answer to this question. I hope you understood very well these questions. Thank you very much.